is a hot topic, quite controversial. I have no conflict of interest to declare, and briefly I'm going to talk about epigenetic marks, genomic imprinting, A.R.T. and possible potential epigenetic disorder, and then I will talk finally about a little bit about cryopreservation and effect on epigenetic with the conclusion. So what is epigenetic? Epi, it's a suffix from Greek old language that means on top of genetic, and basically it's a chemical modification of chromosomal DNA that will basically regulate the expression of genes. So it's a mechanism to make a gene to be expressed or not. So it's a kind of on-off. We can see here the DNA structure as is known, and we know that genes or cluster of genes are associated and responsible for different characteristics in every single individuals. So when we are talking about sequences or about epigenetic, we are not going to change, of course, the DNA structure, otherwise we'll have a mutation, but we are going just to add some specific chemical compounds, which are basically, we can call like little tag. Now it's the time of tag. And these tag are chemical modification, which are mainly methylation, acetylation, phosphorylation, and ubiquitization. So we can see here the methyl group, which is on the DNA structures, and here as well on the histone we can see acetylation as well, which are the main mechanism of regulation. In terms of CH3, so methylation, we have a different enzyme, and there are at the moment 5 DNA methyltransferases. And the CH3 is normally added to the C5 ring of cytosine. Of course, the epigenetic it's regulate, as we say, the expression of genes, and as an impact, and as we've seen also yesterday, they may have an impact associated with pollution, or as Datlio said before, with the nutrition. So in terms of histone as well, it can be modification related again to methyl group, phosphate group, and that can be, as we can see here, when the chromatin is inaccessible or is more accessible here, and this is again with the acetyl group. So basically, when we have a methyl group which is associated with the DNA, that means that no transcription can happen. So the transcription is blocked. So methylation is a transcription blocked. When the methylation is removed, so demethylation means transcription unblocked, and the amount of modification can be they are identified as epigenome. The epigenome need to be specifically and correctly in order to make a viable embryo, and those epigenetic marks during the reprogramming are basically in a way associated during the A.R.T, but not only during the A.R.T, when the 5 days of in vitro embryo development, but even during the gametogenesis and the fetal heart, and we'll talk on that later on. Just a little bit of history. In 1942, the term epigenetic was for the first time studied and investigated by Conrad Hall Worthington at Edinburgh University in 1942. We had to wait a few years, 1975, in order to, from Riggs and Holliday, to propose that for the first time the methylation could be associated with the gene expression. And it's not a coincidence that Bob Edwards actually studied in Edinburgh, and he obtained the PhD in 1955 under the supervision of Worthington. So it's not a coincidence that actually today we are talking about epigenetic and IVF, and these two pioneers were actually correlated, and they studied together in Edinburgh. In Edinburgh, there is actually Edinburgh University there is a gallery dedicated to Conrad Waddington. So if you're going to Edinburgh, please, for your curiosity, just visit. So in terms of epigenetic reprogramming, we need to take in consideration three important marks, which they can be erased, they can be established, and then be maintained. So when we talk again of this epigenetic, in terms of erasure, we need to come back on the primordial germ cells. So it's very early in the early development. And then, of course, there is a new imprinting which is established. This is during the gametogenesis. And then, of course, the famous 5 days of A.R.T. when we culture those embryos in vitro. In that case, they need to be maintained. So this is pictures back to the basic of biology, and we see that the germ cells, they start the migration very early during the development. So the primordial germ cells migrate 4 weeks, 5 weeks to make the genital ridge. And at that stage, as we can see here from the fetal gonad, we already have an imprinting erasure. So that's mean the information of a previous generation, they are erased. And then they need to be established, the new imprinting, so the DMRS, different methylated region. These they need to be established and maintained during the zygote, as you can see here, and then during the blastocyst formation. So this part here is the one which is involved with the A.R.T, the 5 days of in vitro development, but this is not only this one. We need to take in consideration the fetal gonad, so the fetal life, and then all the gametogenesis. That's why very often I had people say, well, when you go to the blastocyst stage, you are going to alterate the epigenetic. Yes, that might be true, but this is not only the steps. We need to take in consideration gametogenesis, spermatogenesis, oogenesis, the stimulation, and then even the fetal heart. So yeah, basically in these pictures as well, this has been reported. So these are the 5 days from the fertilization to the embryo development. But all this part is important as well in terms of epigenetic regulation. So of course, the embryo in vitro is not like in vivo, and there are different parameters that might be alterated, like changing of temperature, pH, air quality, reactive oxygen stress, and antioxidant, which can alter embryonic proteome. And then this is of course important. Another aspect is the genomic imprinting. That means when there are some genes that are only expressed on paternal side or on maternal side. There are not many genes, there are about 150, 200. As we can see here, there are some are expressed on paternal side and some on maternal. These imprinted genes, well, this is the mechanism. So basically, in this top panel, these two genes, they are not imprinted. In the central part here, we can see that the gene imprinted is not expressed. So in this case, it is only the expression on the male. And in this other side, there is the methylation here. So this is not expressed, and it's expressed on the female side here. So basically, those imprinted genes, they are at the moment they are around 150, 200, 60, 70 of these they are very highly investigated, and some of these genes like MAST, H19, EGF2, they are quite important because they are associated with the placental formation and growing, brain function, physiological BV. They can be associated with the brain development and even with the cancer disease. So these are some studies that has been correlated, for example, the imprinting the MAST and alteration which are associated with the single Russell syndrome, for example. This is a study for the group of John Huntress in Leeds. These are 10 human blastocysts which has been investigated with the MAST, the gene, and we can see that the one in red, sorry, are basically relaxed imprinting, so that means they are lost the imprinting, and this can be case of alteration. This is another study which has shown imprinting error, again in H19, SNRP, and as we can see here, there is imprinting alteration in day 3 embryo, which is 66%, and even a blastocyst. So again, this is back on the concept that when you go to blastocyst, you might alterate, you might have some alteration, but this is even on day 3. And this same study, they found some alteration even when using donor sperm, because this is one of the points that very often you say, yes, but this population cannot be compared with the normal population because the parents, they are infertile, so they might have a problem since they are infertile. But if you have even an alteration with using donor sperm, so that means there is something else that is not really clear. So these are the main syndromes which are the genomic imprinting, the Angelman syndrome, Becker-Whitton syndrome, Silver-Russell, Prader-Willi, and maternal hypomethylated syndrome. These are the initial study, the one from Gerald Cox in 2002, but there are many other studies which I'm going to show later on. And again, I mean, this is not, you know, the message of my talk is not like alarming. There are a few studies that have shown there is an increase of this syndrome in baby born in ART, but the message is not alarming. The message, or I want to take your attention on the fact that there is a loss of methylation in this, for example, for Becker-Whitton syndrome in this specific region, which is KVDMR1, and this is the alteration of the gene H19. So this is common in the ART baby which develop this syndrome. So this gene, the H19, which is paternally methylated, so should be not expressed, but in the baby which have this syndrome after ART, this alteration has been found. So this is, and this has been found by many other studies. This is again a different other study. They found an increase of this alteration in the H19 as well in maternal hypermethylation in 70% on the baby born after ART and develop the Becker-Whitton syndrome compared with 5% in general population. These are different other studies that is just for references here. Again, this is a study from Ura, I think it's from Japan, as well as the alteration, this specific H19. This is a review from Odom and Seegers. So again, and alteration in methylation has been found for, this is another study published in 2009, and in breeding disorder, this is also a study from UK from the group of Batlakiava. This is one of the paper published of Nature Communication as well. Again, in this case has been reported methylation error after baby born with the fresh and frozen embryo transfer. This is one of the paper I found from Italy. The region is in Piemonte, which is next to Milan actually. And in this case, these are study in which the Becker-Whitton syndrome has been investigated. As we can see here, they found one affected every 1,000 baby in the RT group, although in the baby born naturally it was one every 12,000. So there is a risk of 10 times. But again, the message is not only the increase of risk, because of course with the number statistician can be discussed, but the point is that it's the loss of methylation that is quite common. Some authors, like Ivan Menezo, for example, has hypothesized that the problem of methylation can be associated with the medial formulation and with the availability of methionine, which is the group that release methylation. So this is something that can be taken into consideration. Now I'm going to talk about the last couple of minutes on the aspect of cryopreservation and eventually association with modification in epigenetics. I'm not going to talk about vitrification in general, but we know that nowadays, in the last decades, going from the slow freezing to vitrification, it's a procedure that is a high use in IVF. We vitrify ovocyte, we vitrify embryo for a variety of reasons. And some of the cryoprotectant we use some sugars like glucose, trehalose, which is important to remove the osmotic gradient, to remove water from the cells, sorry. But some other, I lost. Oh, sorry. Sorry, I was too fast. Yeah, you are. So some of the cryoprotectant we use, like PRH, DMSO, these are the cryoprotectant. They are intracellular, so they goes into the cell to avoid damage during the cryopreservation. And unfortunately, DMSO is one of the dimethyl-sulfox is considered as a high impact on embryo development, but also of cell differentiation, apoptosis, metabolism, and lipid metabolism, and install the acetylation. And there are few studies which have been investigating the effect of the MSO. This is a couple of studies here correlated vitrification with the MSO, which can have alteration in gene expressions. This paper is not, I don't want to create an alarm. This is a paper published recently analyzing the registry from Denmark and Finland, and they found an increase in leukemia in baby born after frozen embryo transfer. There are only 48 cases, and the author conclude that this data should be considered with caution. It's not alarming, but again, we need probably to take this in consideration and to collect more data, maybe to have some registry, European registry of something to assess and investigate it even in the next 2-3 decades. To see if something is really associated. Of course, it's already well known that baby born from frozen embryo transfer, they are large for gestational age, and on the other side, the baby born from fresh embryo transfer, they have a risk of small gestational age. And we know that large weight or small can be associated with the cardiometabolic risk factor in the future. And again, the weight can be associated with alteration in placental formation. So, it's something that we need to take in consideration. These are a couple of papers that have been published from our group, and this is just accepted on publication on JARC for your references. So basically, in conclusion, and I hope to be on time, normal embryo development require correct epigenetic information, and imprinting and methylation can express and regulate gene expression. A.R.T is quite associated, it's a very delicate time when we grow in vitro, and then can be associated with some alteration in methylation on and like a small increase on associations with epigenetic disease. So, of course, A.R.T is safe, is considered safe. There are more than 9 million babies born with this technique, but we need to have always an extra eye to identify which are the best cultural conditions to provide epigenetic stability, and also, to continue to provide an IVF treatment which is minimal and to reduce the risk eventually of epigenetic disease. And thank you very much for your attention.